Yes, ma'am, I've started the recording.
So, welcome back to Translational Oncology Insights. This is the second part of our exploration of cancer cell reversal with Dr. Pathak. In our previous episode, we delved into the mechanistic foundations of cellular reprogramming. Today, however, we are addressing the translational challenges. How do we move from laboratory full proof of concept to the clinical reality? What are the regulatory, technical, and safety hurdles in implementing reversal strategies in human patients? Dr. Pathak, transitioning from mechanistic understanding to therapeutic application, represents one of the most challenging aspects of biomedical research. What are the primary barriers to clinical translation of cancer reversal strategies, do you think?
Thank you, Ritika. So I think the the translational challenge is a multifaceted and a perhaps more complex than traditional cancer therapeutics. This is because we are not just trying to kill cancer cells; we're trying to fundamentally reprogram them while ensuring the reprogrammed state is stable as well as safe. So the primary barriers they fall into several categories. First is the delivery challenge. That is, how do we get reprogramming factors into cancer cells specifically at sufficient concentrations and not for the appropriate duration and for the appropriate duration. So the most reprogramming approaches they require sustained intervention over days to weeks, which is vastly different from traditional small molecules. Second, is the stability and safety concern. We need to ensure that reprogrammed cells they maintain their normal identity in long term and don't have to revert to malignancy. Otherwise, the entire hard work goes to waste. Also, this requires extensive validation to ensure that our current preclinical models they accurately predict this one. Thirdly, the heterogeneity problem. Now, the tumors, they aren't monoclonal population. They contain multiple subclones with different genetic and epigenetic alterations. Now, a successful reversal strategy must work across their heterogeneity or be adaptable to different subclone characteristics. And finally, there's the regulatory pathway uncertainty. Reversal therapies don't fit neatly into existing drug approval frameworks which are designed around toxicity and efficacy endpoints and not the cellular identity concept. That's it.
Right. So delivery systems for reprogramming factors face unique challenges due to the need for very sustained and controlled release, which I guess is very important, and also cell type specificity. So how are you engineering advanced delivery platforms, perhaps using lipid nanoparticles, viral vectors, or biomaterial scaffolds to achieve therapeutic reprogramming in vivo?
So, delivery is arguably our greatest technical challenge so far because reprogramming requires precise spatio-temporal control over multiple factors simultaneously. The traditional drug delivery assumes single molecules with defined pharmacokinetics, but reprogramming often requires cocktails of proteins, nucleic acids, and small molecules with very different physiochemical properties. We're developing hybrid lipid nanoparticle systems that can encapsulate proteins and nucleic acids while providing sequential release. The key innovation is using pH-responsive lipids that destabilize in the acidic tumor microenvironment, providing tumor-specific release. Also, we have incorporated targeting ligands like antibodies against cancer surface markers to achieve cellular specificity. So for sustained protein delivery, we are using engineering protein polymer conjugates that extend half-life from hours to days. Now this is crucial for transcription factors means need to maintain nuclear concentration over days to weeks or time scale required for chromatin remodeling. Also, we are using a site-specific conjugation to maintain protein activity while dramatically extending the residence time. Also, the viral vectors they offer a very unparalleled transfection efficiency, but they also raise a safety concern. So currently, we are developing episomal viral systems, particularly based on adenoviruses, that provide sustained transgene expression without the genome integration. The challenge is payload capacity As most reprogramming strategies, they require multiple factors that exceed single vector capacity. So far, our most promising approach is biometrical scaffolds that provide localized sustained delivery directly within tumors. So we inject hydrogels that polymerize in situ and release reprogramming factors over weeks, to be more precise. This approach provides a high local concentrations while minimizing systemic exposure, addressing both efficacy and safety concerns.
Right. So, monitoring and validating such reprogramming in clinical setting requires very sophisticated biomarkers that can distinguish between genuine cellular reprogramming and transient phenotypic changes. So, what multiomics approaches and imaging modalities do you develop for real-time assessment of reprogramming successes?
This is absolutely critical because transient phenotypic changes can mimic the true reprogramming, but only genuine reprogramming provides lasting therapeutic benefit. So we need biomarkers that report on stable epigenetic state changes and not just temporary gene expression shifts. Our primary approach uses liquid biopsies to monitor circulating tumor DNA methylation patterns. Now, successfully reprogrammed cells should show restoration of normal methylation patterns as key regulatory regions. So, we have developed a targeted bisulfite sequencing panel that focuses mostly on the informative CPG size, essentially the epigenetic fingerprints that distinguish normal malignant and reprogrammed states. A single cell RNA sequencing of circulating tumor cells, it provides a real-time assessment of transcriptional states. Also, we have identified gene expression signatures that are unique to genuinely reprogrammed cells versus those that are simply growth arrested or stressed. The key is looking for regulatory networks and not just the individual gene. Also, for imaging, we are developing PET tracers that report on specific metabolic states associated with successful reprogramming. Reprogrammed cells they often show restored normal metabolism, which creates a distinct metabolic signatures detectable by specialized tracers. Now, we are particularly excited about tracers that report on SAM metabolism since this directly reflects the methylation capacity. Also, advanced MRI techniques, particularly ones with the diffusion weighted imaging and MR spectroscopy, they can detect even changes in tissue microstructure and metabolism that accompany successful reprogramming. Also, these provide non-invasive and whole tumor assessment that complements molecular biomarkers. Now, the most sophisticated approach is integrative biomarker panel that combines epigenetic transcription, metabolic, and imaging readouts. Machine learning algorithms trained on preclinical reprogramming data might help to interpret these multidimensional data sets and predict the long-term reprogramming stability. But existing paradigm. We are working closely with FDA's Office of Cellular Tissue and Gene Therapies to develop appropriate regulatory pathways. The key insight is that reprogramming therapies may need a hybrid regulatory frameworks that combine elements from gene therapy, cell therapy, and traditional drug approval process. Now, safety assessment is particularly complex because we need to evaluate both immediate effect and long-term stability of reprogrammed states. A traditional phase 1 dose escalation studies may not be appropriate because dose isn't just about concentration, it's about combination and timing of multiple factors. So we are developing adaptive trial designs that can explore multidimensional parameters spaces very efficiently. Also, long-term safety monitoring is crucial because the ultimate risk is reversion to malignancy, possibly with new and more dangerous characteristics. So we are proposing extended follow-up periods, potentially 10 to 15 years, with sophisticated monitoring protocols that can detect early signs of reversion before clinical manifestation. Also, biomarker qualification is essential for regulatory approval. We need validated biomarkers that regulatory agencies accept as surrogates for successful reprogramming. This requires extensive clinical validation studies that demonstrate correlation between biomarker changes and long-term clinical outcomes. International harmonization is also very crucial because reprogramming therapies will likely be developed globally. Since we are working with the European Medicines Agency and other international bodies to ensure consistent regulatory approaches that facilitate global development and approval.
So, the economic and manufacturing challenges of personalizing reprogramming therapies are substantial. So, how do you envision scaling production of patient specific reprogramming cocktails, and what health economics models support the value proposition?
The economics of reprogramming therapies are fundamentally very different from traditional pharmaceuticals because they need a very patient specific or tumor specific formulation rather than one size fits all approaches. Now, this creates an unprecedented manufacturing and cost challenges. For patient specific therapies, we are developing modular manufacturing platforms that can rapidly assemble the reprogramming cocktail from standardized components. Also, think of it like a custom pharmaceutical compounding, but with sophisticated quality control and regulatory oversight. We're using automated synthesis and formulation systems that can produce small batches of personalized medicine within days of receiving patient samples. The cost structure is challenging because development costs are spread across smaller populations, but manufacturing costs for those are higher than the mass produced ones. However, the value proposition is compelling if we can achieve a genuine long-term remission of cure. A therapy that costs $5 lakh but provides durable cure is economically attractive as compared to the traditional cancer care that often costs $2 lakh plus annually for palliative treatment. We are developing a health economic model that account for the unique characteristics of reprogramming therapies, like high upfront cost but potentially lifetime benefits. The key metrics are quality adjusted life years and total cost of care over extended periods. The preliminary analysis they suggest that successful reprogramming therapies will be cost effective even at high price points. Value based pricing models are particularly relevant for reprogramming therapies. So we can structure payments based on reprogramming success rates and durability, aligning economic incentives with clinical outcomes. This reduces the payer risk while providing developers with incentive for genuine therapeutic innovation.
Indeed, that was a well-rounded answer. So, looking towards the future, so how might advances in synthetic biology, AI-driven drug design, and precision medicine converge to create next-generation reprogramming platforms? And what interdisciplinary collaborations do you think are very essential for realizing this vision?
The future of reprogramming therapy lies at the intersection of multiple cutting edge technologies, like creating possibilities that far exceed what any single field could achieve independently. So synthetic biology is revolutionizing how we design reprogramming systems. So instead of relying on natural transcriptional cofactors or transcription factors, we are engineering synthetic regulatory circuits that can precisely control and are less likely to have unintended effects. We're developing programmable epigenome editing systems that can simultaneously modify multiple chromatin states with unprecedented precision. Also, the AI-driven approaches are transforming how we design the reprogramming strategies. Machine learning algorithms trained on massive data sets of successful and failed reprogramming experiments can predict optimal factor combinations for a specific cancer type or even individual patients. Also, we're developing AI systems that can design personalized reprogramming protocols based on tumor molecular profiles. Precision medicine integration ensures that reprogramming strategies are tailored to individual tumor characteristics. Now, multiomics profiling of patient tumors guides the selection of optimal reprogramming approaches, while real-time monitoring allows for adaptive protocol adjustments. Now, this creates a closed loop therapeutic system that continuously optimize reprogramming success. The essential interdisciplinary collaboration span for beyond traditional biomedical boundaries. We need partnerships with computer scientists for AI algorithm development, material scientists for advanced delivery systems, bioengineers for manufacturing platforms, and economists for a value-based care models. Perhaps most importantly, we need collaborations with ethicists and social scientists because reprogramming therapies they raise profound questions about the nature of disease, cellular identity, and what constitutes appropriate medical intervention. These technologies could fundamentally change how we think about cancer. From a disease we fight to condition we correct. The vision is ultimately about creating a new paradigm in medicine where we don't just treat diseases but correct the underlying cellular malfunction that caused them. This represents a transition from symptomatic treatment to genuinely curative medicine based on cellular reprogramming. Thank you.
Dr. Pathak, thank you for your comprehensive analysis. With that, we close our podcast, Translational Oncology Insights. Thank you, everyone.
Thank you. Bye.
Right. So basically, resistance mechanisms to reprogramming interventions. In that topic, it likely involves both genetic and epigenetic factors, which are both at interplay. So, how do you anticipate and counteract resistance, particularly in the context of clonal evolution and tumor heterogeneity?
See, resistance to reprogramming is inevitable and multifaceted. It involves both hardwired genetic alterations that prevent reprogramming and adaptive responses that restore the malignant state after initial successful reprogramming. Now, the genetic resistance mechanisms they include mutations in chromatin remodeling complexes that make cells refractory to epigenetic reprogramming. For example, cells with inactivating mutations in SW or SNF components may be unable to undergo chromatin reorganization, necessary for identity stitching. We developed genetic stratification approaches to identify patients whose tumors have retained reprogramming competence. Epigenetic resistance is more subtle but equally important. Some cancers may exist in particularly stable epigenetic states that we call locked chromatin configurations. That is, it resists reprogramming signals. These often involve multiple reinforcing epigenetic marks and altered nuclear architecture. So we are developing a primary strategies that use chromatin remodeling agents to unlock these states before applying the reprogramming factor. Also, adaptive resistance, it involves initially reprogram successfully, but then evolve mechanisms to escape the reprogramming state. This is particularly challenging because it suggests that reprogramming itself creates selective pressure for reversion. We are studying the dynamics of this process using longitudinal single cell sequencing of program cells. Our counter strategies involve combination approaches that target multiple vulnerability points simultaneously. Also, developing adaptive protocols that monitor for early resistance signature and automatically adjust deprogramming combination is in pipeline. The goal is to stay ahead of resistance evolution by anticipating likely escape paths. So the most sophisticated approach is evolutionary steering, using our understanding of cancer evolution to guide cells through irreversible reprogrammed states. This involves identifying epigenetic states that are evolutionary dead ends or malignancies, ensuring that even if cells continue to evolve, they can't easily revert to dangerous phenotypes. That's it.
Right. So, regulatory approval pathways for reprogramming, like cell reprogramming therapies, are largely unestablished. So, how do you envision working with regulatory agencies to develop appropriate safety and efficacy frameworks, particularly with that of regarding long-term safety monitoring?
The regulatory landscape for reprogramming therapies is genuinely uncharted territory. The current frameworks assume that therapeutic interventions either kill disease cells or modulate their function temporarily. The concept of permanently altering cellular identity raises novel questions that don't